in that egg and watch development proceed. This process, the question that this experiment asks is, can a nucleus from a cell that already has a fate or is further along in development, can it go back to an embryonic state and then in that embryonic state specify all the tissues of the body? And in this picture here, we've actually got a pipette which through suction is holding an embryo. And this is another glass pipette which has a tiny little nucleus in it which is being injected into the cell. So the idea to do this actually has been around for a long time. Back in 1895, a French scientist named Yves Delage predicted, now he didn't do this experiment, but he predicted that if you took an egg and took out the nucleus and put in another cell nucleus from another ordinary embryonic cell, which presumably means a later stage of development, he thought that you could actually get normal development to proceed. Now, he didn't actually do this experiment, but Hans Spemann did something similar, or he tried to design an experiment now in 1928. So Spemann is known to most or all developmental biologists as having played an instrumental role in understanding how body patterns are established, and he did win a Nobel Prize for that. But this experiment is not what he won the Nobel Prize for, but it was, I'm not going to go through the specifics of this experiment with you unless you want to know later, but what he did is he was able to form, take a nucleus from an embryonic cell that was at let's say the 8-cell stage, I think it's the 8 or the 16-cell stage, and then allow that to kind of slide into an egg that had no nucleus. And he showed that it could actually form a little salamander that was eventually, this one's not fully formed, but it actually worked. So you could